Hi, welcome to Chapter 6 vocabulary video. Chapter 6 is all about the sort of nutritional needs and environmental needs that microbes have in order to grow. So organisms can get their energy from different sources. Some organisms get their energy from light. Phototrophs make energy from light, so through photosynthesis, essentially. Plants, cyanobacteria, algae, some kinds of fungi can do that. Right? But a lot of the organisms that we will be dealing with that so us, humans, and also most of the pathogens and organisms that live on us get their energy from chemicals, chemotrophs, from some kind of organic material like glucose. Right? When it comes to getting nutrients into and out of cells, there are different ways that chemicals can be transported in the body, but also via cell membranes. So a lot of transport occurs by diffusion. Diffusion is a natural, spontaneous chemical process where molecules from an area of high concentration move to an area of low concentration. Here's my little depiction. All right, so up here there's a high concentration, down here there's a low concentration. So they will naturally diffuse or move down the concentration gradient to the area of lower concentration. Naturally, things want to spread out and increase their entropy. A specific type of diffusion is osmosis. Osmosis is the diffusion of water. It's the movement of water from a concentrated solution to a dilute solution. Nope, other way around. From a dilute solution to a concentrated solution. The dilute solution has more water than the concentrated solution. So water will move from more dilute to more concentrated, which kind of sounds like the backwards version of diffusion, but it's not in the context of water because it's moving from where there's more water to where there's less water. That is osmosis. All right, and osmosis is really important for cells. It affects the amount of water that's in the cell. It is determined by the amount of water or concentration of the environment that cell is in. So ideally, cells want to be in an environment that is isotonic to their internal solutions. They have a certain amount of water and a certain concentration of salts and sugars inside the cell. So ideally, they want to be in an environment that has the same concentrations. And in that case, osmosis is sort of static, or it's at equilibrium. There's the same amount of water moving out of the cells as there is into the cell. If the cell finds itself in a hypotonic solution, then the concentration in the environment outside the cell is lower. Hypo means less than. And in that case, water starts to move into the cell, which could cause it to rupture, definitely will cause it to swell, and it's just generally not great. Hypertonic is the opposite. In a hypertonic scenario, the cell's in an environment that has a very high salt or sugar concentration, so the solution concentration is very high. And in that case, water will leave the cell. That will those solutes draw water out of the cell, dehydrating the cell, which can make it difficult for the chemistry of the cell. It can become too dehydrated. So ideally, isotonic is the best. Iso means same, by the way. So osmosis is a type of, and diffusion are types of passive transport where things are automatically moving. It doesn't take any energy for particles to move from an area of high concentration to an area of low concentration. The other type of transport really is active transport. So in active transport, think it takes energy to be active. So an active transport, you have to push the molecules, and we give them a push by using energy or ATP. So a lot of times you hear there are different cell transporters in the cell membrane. You've got protein channels and then you have protein pumps. The channels are for passive transport. Things just go through them. But the pumps literally push using ATP to push stuff out as a type of active transport. So when you think active, think energy is required. Phagocytosis. Phago, again, means eating. So phagocytosis literally means a process of cell eating. So cells, I like to think of amoeba as the one that I get pictured in my head, but cells can actually scoop up bits of their environment and take them into their cell, and it's kind of considered cell eating. Phagocytosis is the process of cell eating. They can also do penocytosis, which is the process of cell drinking when it's liquid. Optimum temperature. This is just the best temperature for a bacteria or microbe of any kind to grow at. So microbes, let's say E. coli that we work with in the lab, it can grow at a lot of different temperatures. It'll grow in the refrigerator at 4 degrees Celsius. It'll grow at room temperature on the desktop at 25 degrees Celsius, but it grows best at 37 degrees Celsius. And there are ranges where it won't grow. So the optimum temperature for an organism is the temperature where it grows best. It's not the only temperature it grows at, just the one where it grows best. So organisms that grow best at colder temperatures, we call those psychrophiles, meaning they like the cold. Organisms that grow best at sort of intermediate temperatures and our body temperatures in that range, so like room temperature, body temperature, are both would be in the mesophilic range. So those are called mesophiles, middle range likers. And those that like really hot temperatures are thermophiles, and there's even hyperthermophiles too, that like they can grow in boiling hot scenarios. All right, most human pathogens are going to fall in this category. They're mesophiles because that's where our body temperature falls. So they're going to be organisms that grow optimally at our body temperature. Some other conditions that bacteria may like or dislike. Salt is usually something that inhibits growth of bacteria because it draws water out of the cells. It's hypertonic. All right, but there are some microbes that have very high salt concentrations and have evolved to like high salt, and they are called halophiles. Halo, as in halogen, chloride, bromide, fluoride, iodide, those are all halogens, so salts. Halo means salt. Symbiosis is a process. Sym means together, and biosis, to live. So it's a process of things living together. Usually when they do that, it's a beneficial relationship, but not always. One of the most beneficial symbiosis would be mutualism. Mutual means both, I suppose. So in a mutualistic relationship, two organisms living together both benefit from that association. So for example, we have bacteria in our guts that produce vitamin K. That is beneficial to us. Our bodies need vitamin K to create clotting factors for healthy blood clotting, and we provide the bacteria with a safe home to live and nutrients from our diet. So that would be a mutualistic relationship. Commensalism is a relationship where one of the partners benefits and the other one just, is neutral. They don't benefit, but they are not also harmed. So a lot of the bacteria in our body are referred to as commensal bacteria because for a long time biologists just thought that they were freeloaders, just living on our body, not causing us illness, but also not helping our health. But it's coming out with microbiome research more and more that a lot of the bacteria in our body are actually mutualistic, that without them we do develop disease. So far more of our normal flora are probably mutualistic, but some are also probably commensal, just freeloaders. Parasitism is a relationship where one of the partners benefits and another one is harmed. So pathogens are parasites. They do harm the host. A lot of helminths. Helminths are all parasites. They grow while harming the host. Antagonism is a competitive relationship between organisms. And this is actually really the root cause for evolution of antibiotics. Antibiotics are actually natural substances that are produced by microbes to help them sort of fight and compete with other microbes in their environment. So think of the soil. There's tons of bacteria and fungi that live in the soil. And in order for them to be able to share resources or compete for resources, they have to have a way to keep the other species at bay. So they produce these substances that kill other species of bacteria or funguses. So antibacterial production by bacteria and funguses is actually a classic example of antagonism. I like to think of it as like bacteria and viruses kind of elbowing each other, like, get out of my space, this is my space. Antagonism. Quorum sensing is how bacteria use chemicals to talk. It's like bacterial sign language in a way. So quorum means, I don't know the exact definition of it, but it means to have like a group of people that meets a threshold. So like Congress, I think, might have one, like different religious groups have quorums for like prayer, or government groups will have them for meetings. You have to have a certain percentage of the governing body there in order to vote, okay? So with bacteria, quorum is it's how bacteria sense how many other bacteria are around them. They can't see, they can't hear, they can't talk, but it does affect their ability to survive if they have 2 other like bacteria or 2 billion other like bacteria around them. And they actually have a way to communicate their population sizes by secreting chemicals, and when those chemicals reach a high enough concentration, it actually turns different genes on. And it basically triggers this population count. That's called quorum sensing. So quorum sensing is sort of like bacterial census counting using chemicals. Biofilms. A lot of times, quorum sensing leads to biofilm formation. When there's a lot of bacteria around, they can stick together and form these thick growths that are almost like tissues. I mean, they're pretty advanced and they work together. And biofilms are really problematic in medicine because they're much more difficult to treat. They usually need to be physically disruptive. You can't just give somebody antibiotics because it won't penetrate the biofilm. It's sort of a thick layer of microbial growth, oftentimes containing multiple types of microbes, not just one species. Microbes divide by just literally copying and splitting themselves in half, and that process is called binary fission. And when you are culturing bacteria and they're growing and they're doing binary fission, you can sort of chart their growth in this something called a growth curve. And there's 4 parts of the growth curve. So when you inoculate a culture, the first hour or 2, there's a lag period. So the bacteria are doubling from like 1 to 2, 2 to 4, 4 to 8, 8 to 16, all right? These are pretty small changes initially. But then those numbers start to get really large. And so each time you double, you're making logarithmic changes in growth. And so that is the log phase. Well, I guess the doubling that I just described is part of the log phase. The lag phase is really more like maybe they're not quite doubling yet. They're just kind of getting used to this new environment. Maybe they're waking up. They were in the fridge before at not an optimal growth temperature, and now they are at an optimal growth temperature, so they're just starting to kind of wake up. Then when they're doubling, that's the log period. But eventually the culture is going to get so full of teeming growth of bacteria that they start competing with each other for resources. The nutrients start to run out. They're getting crowded for space. So they stop growing. And then they actually start to die because they get old, and also the nutrients start to run out. So this is the sort of growth curve that you get when you're growing bacteria. So when I inoculate a culture for lab, I do it and I trying to catch bacteria in this log phase, maybe the stationary phase for us to use them. I don't want to inoculate the cultures so early that by the time we have lab, they're all in that death stage of growth. Turbid is the word that basically just means cloudy. So when a broth culture becomes cloudy, that's a sign of bacterial growth. Sterile broth culture is clear, transparent, you can see through it. But turbid, when you shake it up, you can see little particulates, or you might not even need to shake it, you can just look at it and it's cloudy. So the word for that is turbid, and it's a sign of bacterial growth. And that is the end of Chapter 6, Vocab.